So today we are continuing with genetics. In the next portion of genetics, we are going to find out by certain experiments which proves that DNA is the genetic material. So there are a series of experiments performed by scientists. The first experiment was performed by Griffith, also known as Griffith's experiment, in which he proposed the transforming principle. So in order to understand his experiment, first there are a few things you have to know, like pneumonia is caused by Diplococcus pneumoniae. This bacteria exists in two strains. Rough strain and the smooth strain. Rough strain does not cause pneumonia. Smooth strain, it causes pneumonia. So two strains of bacteria, Diplococcus pneumoniae. This is the bacteria which causes pneumonia. It exists in two strains, rough strain and smooth strain. Rough strain does not cause any pneumonia, smooth strain causes pneumonia. So what happens is, when these were injected into mice, this mice, they survived. They did not have any effects of pneumonia. But whereas these mice, when they were injected with the smooth strain, they all got pneumonia and eventually died. So smooth strain causes pneumonia, rough strain does not cause any pneumonia. Now what happens is, again, in the second case, heat killed rough strain and heat killed smooth strain were injected in mice. Again what happens is, mice survived, and in this case also the mice survived, because now this strain is no longer living because it has already been destroyed by heat. Now in the final case, what happened was, living R strain plus heat killed smooth strain were injected in mice. What happens is, the mice showed symptoms of pneumonia and they died. So from this experiment, Griffith concluded that there was something responsible in which this heat killed smooth strain transformed the rough strain into a virulent form. That means this was able to cause pneumonia. So this is also known as the transforming principle, because heat killed smooth strain was able to convert the rough strain and cause pneumonia, which led to the death of the mice. So this is an experiment by Frederick Griffith, also known as transforming principle. But this experiment is not conclusive, because Griffith did not conclude as to what actually caused the transformation. He only said that something must be responsible in heat killed smooth strain that converted the R strain from non-virulent to a virulent form. This is what he concluded. He did not conclude as to what actually caused the transformation. So in order to understand this, there is another experiment performed by McCarthy, MacLeod and Avery. So what they did was, heat killed S strain plus rough strain. These were subjected with a series of enzymes. One such enzyme was DNase. DNase is an enzyme which breaks DNA. So what happened was, the mice did not get any pneumonia. That means mice lives. It's the same experiment as the previous one, just that in addition to the heat killed S strain and rough strain, what we actually added was DNAs, not we, Avery, McLeod and McCarthy. So the mice lives. Now again the same was injected with another enzyme. Protease is an enzyme which breaks proteins. In this case, what happens is the mice die. So from this it proves that DNA is actually transformed into a rough strain. Because since we are using protease, protease breaks DNA, it does not affect any DNA. But whereas when we were using DNAs, it destroyed DNA, so we could not observe pneumonia. So these are the two experiments which prove DNA as the genetic material. Another conclusive experiment was performed by Hershey and Chase, also known as the Bacteriophage experiment. So a bacteriophage is a virus which consists of a head and a tail. The head contains the genetic material. Bacteriophage is a virus which infects bacteria. So when it infects a bacteria, what happens is only the genetic material enters the bacterial cell, and from this genetic material you get many more bacteriophages. So bacteriophage is a virus which affects bacteria, and only the genetic portion of the bacteriophage enters the bacterial cell. Now your head is made up of protein, and the genetic material obviously contains DNA. Now DNA has phosphorus and protein has sulfur. So what Hershey and Chase did was he grew this bacteriophage separately into medium containing radioactive sulfur and radioactive phosphorus. So when these bacteriophage were grown in medium containing radioactive sulfur and radioactive phosphorus, what happens is the bacteriophage that you gain from this medium shows radioactivity for sulfur. This also shows radioactivity for phosphorus. But the thing to remember is, since phosphorus is present in DNA, this bacteriophage shows radioactive phosphorus in the inner side, but whereas on this, radioactivity is observed on the outer side because protein contains sulfur and your head is made up of proteins. Now, in the next step, what they did was both the bacteriophages these were allowed to infect a bacterial cell. Now earlier I told you When infection occurs, only the genetic material enters the protein coat, it never enters. So what happens is, in this case, both the DNA entered. So now what happens is, this bacterial cell produced many bacteriophages, and the bacteriophages, what they did was, they showed radioactivity for phosphorus. So this proves that genetic material is basically made up of DNA, because in this case, since the protein coat, it never got to enter, it was never transmitted or inherited in the next generations, but in this case you got radioactivity. So that means DNA is the genetic material. So all these experiments provide evidences for DNA. The first experiment was transforming principle by Griffith. The next experiment was transforming substance by Avery, MacLeod, McCarthy. And the third experiment was Bacteriophage experiment by Hershey and Chase. So these three experiments prove that DNA is the genetic material. The thing to remember is, in transforming principle, it was not conclusive. They only concluded that something must be responsible in heat killed S strain, which transformed the rough strain into a virulent form. From this experiment, it was proved that DNA is the genetic material, because they performed the same experiment but slightly with the addition of different types of enzymes. They added DNase, they added protease. When they added DNase, the rough strain was not converted into the virulent form, because DNase destroys DNA. But when they added protease, they obtained virulent rough strain, because protease destroys protein. So from this it was proved that DNA is the genetic material. And finally, from the bacteriophage experiment, Hershey and Chase, what they did was, they found out by using radioactivity for phosphorus and sulfur. Phosphorus is present in the DNA, sulfur is present in the proteins. So that means radioactivity for phosphorus was obtained in the coming generations, which is transmitted or inherited. So this obviously proved that DNA is the genetic material. So from the previous chapters, we all learned that certain factors called genes are responsible for characters. So basically, when we talk about genes, it can either be a cistron, muton, or recon. These are all units of genes. By cistron, what it means is a functional unit which codes for proteins or amino acids. Mutons means a segment of DNA which can undergo or produce mutations, and recon means a segment of DNA which can produce new combinations by undergoing recombination. nations. So this is basically what it means by genes. So when we talk about genes in details, there are many things like a gene is responsible for producing an amino acid. An amino acid codes for polypeptide and polypeptide codes for proteins. So in the next class, I'll be discussing about protein synthesis.